that's maybe rich in carbohydrates but doesn't have a lot of proteins. Well, then the bacteria are not going to be able to get any tryptophan from their environment. So they got to manufacture it for themselves because all cells need all the 20 amino acids to make their proteins. But that's not a big deal because bacteria have enzymes for manufacturing the amino acids if they can't get them from their environment. And these illustrate the enzymes. And so these enzymes are going to be a team of enzymes that manufacture tryptophan for the bacteria when the bacteria can't get it. And remember the term here, this is a metabolic pathway. A metabolic pathway is a group of enzymes that working together can manufacture some molecule, because usually one enzyme is not sufficient to generate a molecule all by itself. So, for example, maybe the starting material, the substrate for this pathway is a carbohydrate, but each enzyme makes a small change in that and then passes the molecule off to the next enzyme until we finally get down to the tryptophan being manufactured. So this is fine for the bacteria. If you eat carbohydrates, it can still make the amino acids that it needs. But notice that the bacteria had to expend some energy and resources to make this metabolic pathway. It had to expend ATP and nucleotides to express these genes, to make the mRNAs, and then it had to expend ATP to have the ribosome link together the amino acids, and it certainly had to use amino acids to manufacture these enzymes. So it's not a free lunch for the bacteria. It definitely has to invest resources to make this metabolic pathway to generate the molecules that it needs. And so it really only wants to make this metabolic pathway when it needs to, when there's no tryptophan in the environment. So in other words, if suddenly you start eating a protein-rich meal again and tryptophan does become available from the outside, it wants to stop expressing these genes, because as I said, those would be wasteful. That would be wasteful, and so it needs to shut down expression of those genes. But then again, transversely, if you stop eating proteins and there are no amino acids, then it needs to start up expression of those genes to bring back the tryptophan making pathway. And so that's really what this whole section on operons is going to be about. It's how the bacteria can turn on and turn off groups of enzymes that form a metabolic pathway to coordinate their expression, so they're either all being expressed or all being not expressed, depending on the bacteria's needs. And it gets kind of detailed and convoluted, and I'll warn you that some of the concepts here are going to require a little bit of thinking about and reviewing before the exam. But in the end, they do make sense. So just have your thinking cap standing by. That's one thing I'll warn you about. And the other thing is, what I'm showing you about how bacteria do this, they use something called an operon, and you do not find these operons in eukaryotes. We do not have any operons, so we are not able to regulate our gene expression in the same way that I'm showing you here. It's only a bacterial thing. Okay, so what is this operon? And remember the goal again, it's ways of regulating the expression of all the enzymes in the pathway. Well, those enzymes, of course, come from genes. And so somewhere on the bacteria's chromosome, which is this blue line right here, if we look, we should be able to find the genes for those enzymes of a metabolic pathway. And there they are. Gene number 1 makes this enzyme of the pathway. Gene number 2 makes that enzyme of the pathway. And gene number 3 makes that enzyme of the pathway. And notice something interesting here. They're all clustered together, 1, 2, 3. And that is different from eukaryotes. Remember, we eukaryotes have many different chromosomes. We human beings have 23 different types of chromosomes. And what you find in eukaryotes is the genes are not clustered together even if the proteins function in a metabolic pathway. For one of your metabolic pathways, the gene for the first protein of the pathway might be here, the gene for the second protein of the pathway might be here, the gene for the third protein of the pathway might be here, the fourth gene of the pathway there, the fifth gene, sixth gene, seventh gene, like that. They're scattered all over the chromosomes in kind of a willy-nilly random manner. And yeah, the proteins, once they're manufactured from those genes, will function together in the cytoplasm, but the genes themselves are scattered all about. But in these bacteria, just the opposite. The genes of a metabolic pathway are found 1, 2, 3 clustered together in the same region of the chromosome. And here where things really get a little wacky. There's only one promoter for all 3 of the genes, or all the genes in the pathway. And that's really different because in eukaryotes, each and every gene always has its own promoter. You never find several genes controlled by a single promoter. And so that's the essence of it. That's how this bacteria can turn on or turn off all the genes of this pathway simultaneously, because they're all under one promoter. And that thing there has a name. It's called an operon, a region of the bacterial chromosome that contains several different genes, usually for one metabolic pathway, but they're all under the control of a single promoter. So when the RNA polymerase, which transcribes the genes, binds to the promoter like this, it makes one huge mRNA that can give the information to the ribosomes for all 3 of these proteins. In other words, there's going to be a start codon right here in AUG and a stop codon there, but then the ribosome can keep chugging along and find a new start codon here, and so start making protein, and the ribosome will find a stop codon there, and then the ribosome will keep chugging along and find a new start codon there, make protein, and then a stop codon there. All right, so this mRNA then will be manufactured. These genes will be expressed when there is no tryptophan, because that's when we need to make these proteins for the tryptophan making pathway. But when tryptophan is present in the environment, we don't want to express this mRNA because we don't need those enzymes. So if there is tryptophan, no expression of the genes of the operon, and therefore the enzymes made from that mRNA are not made.
Eukaryotes, they regulate gene expression through the RNA.
In eukaryotes, the expression of each gene is also regulated mostly at the transcriptional level. Like if the eukaryote decides it wants to express this gene, then the key step is making the mRNA. And so that is kind of the same as here. But what I'm saying, the difference is eukaryotes, each gene is under its own promoter, controlled by its own promoter. Whereas in this, you find several genes under the influence of one promoter. And as a result, you get an mRNA that encodes, one mRNA might encode for 3 proteins. Whereas in eukaryotes, each mRNA only comes from one gene, and it only encodes one protein. Okay, so let's drill down a little bit deeper into this concept. What the bacteria wants to do is this: it wants to control expression of this operon depending on whether tryptophan is present or not. And when we say control expression, what we mean is transcription. The bacteria wants to, in some cases, have the RNA polymerase bind to the promoter and express those genes. And in other cases, like if there's tryptophan in the environment, it doesn't want the RNA polymerase enzyme to bind the promoter and express those genes. And the way the bacteria does this is with something called a repressor protein. A repressor protein essentially can block the RNA polymerase from binding to the promoter. Here we go. It is a protein that can stop transcription by blocking the RNA polymerase from attaching to the promoter. The repressor proteins do this by locking onto a part of the promoter called the operator. The operator is a sequence within the promoter that the repressor protein can bind to. And so when the repressor protein is bound to the operator, that's going to physically block the RNA polymerase from binding, and that therefore the transcription is going to be off. But when the repressor protein is not binding to the operator, then the promoter is open, so to speak, and transcription will be on. Let's see that. All right, so within the promoter, part of the promoter is this operator sequence, and the repressor protein has a binding site. Somewhere at the bottom of the repressor protein is a binding site where it can lock onto the operator. Click like that, and as long as that repressor protein is sitting there, the RNA polymerase can't attach itself to the promoter, and so we do not get expression of those genes in the operon. But when the operator is not, when the repressor protein is not sitting on the operator, I didn't want to show that part. Go away. Anyway, when the repressor protein is not sitting on the operator, then the RNA polymerase can bind to the promoter, and that's when you get expression of the genes. All right, so let's drill down to the next level. What is it that to control expression of this gene, the bacteria has to be able to control when the repressor protein binds to the operator and when the repressor protein is not binding to the operator. In other words, if it wants to turn off the gene, the repressor protein has to bind to the operator, and if it wants to express the gene, the repressor protein has to not be binding to the operator. I shouldn't say express the gene, I should say express the operon, the cluster of genes. All right, so here's the way this works, is that the repressor protein's ability to bind to the operator can be allosterically regulated. Remember, allosteric regulation means a molecule can plug into the back of the protein, of the repressor protein, and change its shape, and that's going to affect its ability to bind to the operator. And so for this TRP operon, this tryptophan operon, here's the way it works. Tryptophan molecules, like you see here, are the allosteric regulator, and they activate the repressor. And what I'm saying there is the repressor's natural state is that it does not have an open binding site for the operator. So its natural state is to not block transcription. It's not attached to the operator. But when tryptophan is present, the tryptophan binds to the repressor protein and allosterically changes the binding site on the repressor protein to make the binding site open to binding to the operator. Like that. And so now, with the help of the tryptophan, the repressor protein has become activated, binds to the operator, and now that's blocking transcription. And the term for that type of thing that's happening here is co-repressor. You would say that the tryptophan is a co-repressor. It's a small molecule, usually the end product of the metabolic pathway we're talking about, that activates the repressor. By activating the repressor, you turn off transcription of the operon. I think what confuses students about that is the tryptophan, in this case, is activating the repressor, but by activating the repressor, you are unactivating the operon, so to speak. You're blocking the operon. So it's sort of a mental double thing. By activating repressor, you are inactivating expression of the gene, so to speak. Okay, well, I think I'm near enough out of time, and I've probably confused you all already enough. So I will see you guys in lab in a couple hours. Remember to bring your DNA technology handout, please.